you'll get the idea. And also I will record everything once you have all the commands and everything, so you will get your documents. This recording is just for a reference purposes. Later on I will record it so that you don't get any issue with voice or anything like that. It will be very straightforward. I'll try to make a I will have a presentation so that all the steps and everything is mentioned in that video. But today I really want to be pretty much sure that your Linux is responding to the commands which we are going to do it. Okay. So in our previous session, what basically we did was we have just covered the uh, theory part. And why that is important? Because theory part is somewhat connected to our practical part, because a lot of the terms which we use are certainly it's derived from what exactly we want from that experiment sample. Now when it comes to the practical part, we need to do certain analysis. So we are here for the analysis, and the data which we are interested in is uh, RNA Seq data, and we are basically talking about mRNA. Why? Because these are the one who are responsible for the functionality. And when we are talking about the functionality, we are very much interested in the gene expression. Why? Because gene expression is something which is providing us, giving us the list of the genes which are somewhat related to how, in particular sample, how the genes are expressing. So they can tell us about. Yes. Now it's fine. Okay, that's good. Now it's fine, right? Okay. So this is just a brief that basically what we are looking into. We are looking for a gene expression data where we are basically comparing. And why we want to compare it? Because we just want to see that what are the like difference between the genes which has been present in two or more conditions. Okay, because that is very important, because we are not interested into a similarity. If a similarity is also there, we also want to see that how much that similar gene is expressing, like where exactly difference is lying on. That is what we are interested in. Now this kind of analysis are pretty much known as uh, transcriptomic analysis, because obviously we are looking for mRNA. They are also known as the technology in terms of it also known as bulk RNA analysis, because you are taking the whole transcriptomics which are present into your sample and you are just comparing it. And it is also known as your RNA Seq analysis. So any name you will see, largely you will get like transcriptomic analysis, RNA Seq analysis, or a very known name is differential gene expression, differential gene expression analysis. Now that is something is largely known. Okay. Now why this is important? Because whenever you are searching for any paper of your interest, or if you really want to have your data, you will come across with these kind of stumps. Okay. So now when we start with the analysis, so we need certain thing. Okay, for the analysis purposes we need two things. We require a data, because the analysis depends upon that data, that is a raw data, and it should have the the format is FASTQ format. It means that it should have the sequencing information that at this particular position in the sense in the fragment, the position, not where exactly it is present in a genome. Right now we are not talking about genome, we are just talking about with respect to that if unknown fragment is present, that machine should be capable of reading the information that at this particular position this is present. Okay, and that format is saved as FASTQ. Okay, we will look into that files details later on once we are sort of done with our practical part. Okay, and the another thing which we required is tools. Now the tools, obviously, because we are using a dry lab bioinformatics, so we require a software, a tools. So we are using a direct tool which we are interested in, and you will see that these are my direct tool. This is a tool I'm using it. Okay, plus what happens that each tool is made up of different languages, so they have different dependencies which is required. Now why I'm telling you all this thing, because what happens that even when you install R, which I'm going to tell you in our future sessions, so any tool always work with a lot of dependency, something which is required for that tool to work, right? Now these dependency, because of these dependency, if certain dependency is missing in your system, you will get an error. So that's why I'm telling you that dependency is important. How we'll get to know about the dependency, uh, that is depends upon the error which we are getting it. Okay, so whenever you are getting an error, you need to look into the error. I will tell you how to look into the error, and if it is mentioning that this particular package is missing, it means that we need to download that dependency. So this is just for your reference, and it is very important. I will try to use the tools which doesn't require us to individually download the dependency, but I don't have any idea that what might happen in our future. So that just understand that if you are getting any error, there are chances that you might missing certain dependency. Okay, so this is how basically the analysis works, data plus tools. Now first thing what we need to do is that we need to have a data. Okay, that is must. So now the next thing which we are going to do, that we are going to search for a data of our interest. Okay, uh, yes, transcriptomic analysis also meant same only, because transcriptomic is basically you are going for RNA analysis. Okay, now again it depends upon it that what basically they are saying, that whether they are referring uh, in RNA, like in DNA we have coding and non-coding, right? In DNA we have two part, we have coding and we have non-coding, right? So when a person who's doing everything including coding and non-coding, then we say that that person is doing whole genome sequencing, that he is considering everything. If that person is only working with a coding thing, then we say that that person is doing whole exome sequencing. Sorry for that, whole exome sequencing. It means that they are only considering the exome, the coding part of a DNA. Now in RNA, we also have coding RNA, we have non-coding RNA, plus we have a small RNA. Okay, if a person who's doing coding, non-coding, means considering the everything, then it is known as whole RNA analysis sequencing, or whole transcriptomic analysis, like home transcriptomic sequencing, or a whole RNA. So you are getting whole RNA data. Okay, everything including. when you are working with only the coding part, and coding part is basically your mRNA, right? So in that case it is known as whole transcriptomic analysis. So right now we are interested into mRNA, so that's why we are doing transcriptomic analysis. And if you are interested into the small RNA, so you are doing the small RNA analysis. Clear? Okay, so now we need to get a data. So how to get a data is very simple. We have a beautiful, we can say, very convenient database where we can find the data. Okay, one way is that uh, you you might be referring to any research paper. In that research paper you might get the GeoID. Okay, if you are getting that GeoID, then you can go to Geo database and search for it. uh, okay, if you are interested into the cancer, we will look into that. Yes, not an issue, but again I'll show you that how the data size and everything works. If that works for you, it's okay. uh, One data I'm going to choose. If you are interested, for an example, more into cancer and you don't want to tell me, it depends. Okay, so you can search the data on your own, because I'm going to tell you that what is the process for your assignment or just for a practice purposes. Okay, so step number one is that if you're referring a research paper, in that research paper if they have mentioned any ID like a geo ID or accession number, then you can directly retrieve that data. Geo ID or SRR ID or ERR ID, anything. uh, if if you are referring a research paper, and if you really want to replicate that paper, or if you really want to use that data for your analysis, because for whosoever has published a paper, the timeline, the tools might be different, right? So you can take the, if that data is publicly available, you can use that data for your publication. So there is no until unless you are taking the permission, citations, everything, that the normal thing which we do. Okay, so this is a one way, simple way where you have all the information with you. This is a one way. Okay, another way is that you really have no idea and you really want to start from a scratch. Just look into the data. If that data description is good, then you like, okay, why don't I try it? Okay, that is another approach. So this database is very much similar, uh, like Google. Okay, so in the same way, what we can do, that we have multiple options to search. First is that we can search if we have an idea ID, that is a good thing. Another is that keyword, any keyword. For an example, right now you are interested into the cancer, right? So what you can type is that you can type cancer. Simple. Just look for the cancer data and then enter it or search it. So once you do that, you will see the page, the result. You can see that these many entries of the sample or the data is present in the NCBI SRA. Up to here clear? Any doubt? Okay, it's clear. So you will go to NCBI SRA, you will search by simple keyword. If you do not have the accession ID, you will just search simply. So in your case, you are interested into cancer. Now we will do some filtration. Okay, so filtration can be done from right hand side and left hand side. Okay, so let's look for the most important filters which we use to minimize the result and look for the data which might interest us. Okay, so the very first thing which we want is that it should be publicly available. We don't want any data which has some restrictions from its owner, right? So first thing is that whether we can access that data, yes or no. So for that you will click into public. Okay, once you click into the public, you will see the numbers has been reduced. Okay, another filter which we use is basically that the you are interested into RNA, so you will click on RNA. The source is RNA, so you will click into RNA. The another thing is that most of the time when you are going for transcriptomic analysis, you always ended up with parent data. I have already explained you in a previous session what is single end and parent. So RNA mostly they use parent data. So you will search, you will click into parent. Okay, so these are the three most important filtration you might use. Okay, library layout, it depends upon you whether you really want to go with the single end or parent data. So most important thing, yes, you can do it, not an issue. The up to certain point all the data will be different, but at the end when you are doing comparing the data set, that time you can, because at the end you will get a result which is present in a single Excel or single file. Based on that, the RNA R language is used for the comparison between the sequences. You will also see that one experiment will have samples multiple samples, and then at one point you will combine everything just to compare it, because if you want to really look for a difference between, at one point you have to compare between the samples. So you can go for it if the way they are doing it. For an example, if you are interested in 5 different cancers, for an example, now obviously one person might not have the cancer data same, right? But you have found that there are 5 different cancer studies are present in your data. So your point is that in spite of going for a one experiment study, you want to do the analysis for 5 experiment study. So you will do analysis for cancer 1, cancer 2, cancer 3, and then later on you can combine it. Yes, 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 yes. Yes, that is how. In RNA, it is always be a comparison. You will see the data which we are going to select. It will always have something different. One will have a treatment, one will have not treatment. So based on the normal, you compare the things, right? So that is what we do. That happens. So in experiment, you will see that the result we are going to search, it will have more than two conditions, minimum 2 conditions, and then it can go up to 8, 10 conditions. So over here what you are mentioning in a chat, you are giving 2 conditions, a normal, yes, data set is having multiple samples with you. So whatever you have mentioned me that one data set may contain normal sample, another, now normal sample and the cancer sample is comes under the one experiment only, because that scientist might be interested to know that what is the difference between uh, the genes which are expressed in a cancer sample with respect to the normal. So it is a part of one experiment which have many samples within it. Many sample means many conditions they have applied in order to understand uh, the main thing which they are looking into. So in your case, the main thing is that to find the list of genes which are expressed in a treatment sample. Clear? Up to here, clear? Yes, you can do it. You can combine it. Not an issue. You can combine it. You can take multiple datas. Not an issue. You can combine it because at one point when you do the practical part, which I'm going to tell you, uh, one part is something is that where you will combine your data just to see that how differentially genes are expressed in that particular condition or those conditions. Only thing which you need to make sure is that uh, which organism you are working with. So for an example, if you are working with mouse and human comparison, then you should have a reference genome of both. Okay? Yes, it is possible. We'll look into the data and you will again get this thing. There are people who combine all the datas, not an issue. At certain point, we will use them as an individual, and after once we get all the result, then we combine them. Okay, so let's look into cancer. So up to here, I think so you are clear that we are interested to access the public data, we are interested in RNA data, and we are interested into the parent data. Up to here, clear? Yes. Nice. Now again we might have something in our mind like which taxon we are interested in. So in your case, I know that you are mostly very much interested in the human. So then you will select human. Okay. So now whatever whatever the results you are getting the data which is publicly available, which is RNA type, which is a parent, and which belongs to human. Now in case if you don't want anything, then you can clear those filters. Very simple. Now you can see that you have so many entries. So you have 16,039 like sorry 1,63,916 entries you have got. So initially you were getting too many numbers, but now it has been reduced to this one. So now if you scroll down, you can see so many entries. So for an example, I'm just clicking on the this data. Okay, or let me do one thing. Let me just open this in a new tab. So I have opened this particular result. So they mentioned that this is a RNA Seq for human, which we have already searched, and they have PBMC from healthy volunteer. Okay, if you just scroll down, you will see that this is the design they have used it, the total RNA library. Now it depends upon them, the person who's providing this data, like what all information they provide you. Okay, so in this study you can see abstract. If you click into that, they might have given you a very simple one line or two line abstract, or they might have given you a very detailed abstract. That is again depends upon the owner. Okay, so over here what we can understand that this is a RNA seq in the study. It is a RNA seq for human endo material cancer cell line. So they have performed on a cancer cell line, and you can also see that they have compared the control media with the bacteria supernatant treatment, PBMC. So this is the experiment they have did it. Then you can see that under the sample it belongs to human, which we have already known. They know that the source is transcriptomic because we are interested into mRNA, and you can see that the accession ID, and you can see the number of bases, size, and when it got published, like when they have did any changes to the NCBI. So this one is very recent one. This is 26 July only. They have published is not the issue because the data is basically publicly available. It already published over here, but that doesn't mean that you cannot use this data. That's why we have selected a public thing, right? So you will find a papers also online that how I can, because it's the secondary data, you can use it for a publication. You just need to look into the paper which they have did it. You have to just to give a credit to that person, the data you have used it. So you can publish it. Not an issue, because whatever the person has did it, the parameters might be different. The tools might be different. Uh, these data is available to us that so that we can use this data for our kind of analysis, and then we can publish it in terms of reviewing this one or redoing those one. So there's no issue with using this data for the analysis, because nowadays people use secondary data for their publication. Whatever he or she has mentioned in their paper could be the entirely different from what approach you are using it, right? So that's why secondary data can be used for publication. You can also search online that how I can use secondary data, NCBI SRA data for a publication. You will get the help. Papers are available. Okay. Okay, because many of them who publish from us, they use secondary data. So now this is a experiment. Now obviously this is only one sample. They will have multiple samples. Okay, so if under the study, if you click all experiment, under the study, if you click all experiment, you will see that there are 8 items, like comparison. So you can see that this is from the healthy volunteer, healthy, and they might have changed something over here. Some difference might come. Maybe the tool will change or something like that. Maybe the cell line will change, something like that. So we'll see that in one simple experiment. If you just click into that, it will have multiple samples beneath it. Okay, now under the study itself, if you click all runs that you are interested into to find out that how many samples are associated with this particular experiment, if you click over here all runs, you will find, you will open new page. You will see there are 8 samples, the bytes and the bases, and you will see that these all are the accession number. Now these accession numbers are very important, because when we have to download our data, we need to see. You can see that this in this one they have took the blood from different volunteers. So volunteer number 1, 3, 4, 5. So these are the different volunteer, they have used it, and they have used different libraries. These are the library name, the sample name. So they have Na1, Na3, Na5, and you can see that these are the control media, and these are the bacterial supernatant. So now what they want from here is that they want to compare the differential gene expression between the control and the bacteria supernatant to understand the cancer data. Clear? Up to here clear? So for our analysis, what we need the accession numbers. So over here you can see that these are the size of the data, which is pretty big in size, because right now they are in the zip format, but again the size will increase when we download it. So these are the accession number you need it, when you want to download a data. So this is a one way of doing it, getting all the accession numbers, because at the end you need the accession number. Now if this experiment interests you, we can use this data, but make sure that the analysis which we are going to perform, it's pretty big in size. Okay, in a same manner you will search for the another data. You will look into that. For an example, this is another study that HEK cells treated with siRNA. So I'm not interested into siRNA because this is a knocked out one. But if you are interested to see that how basically RNA expression play an important role, then you can see that. So what they have there, they have knockout it. Again, this data can be used, not an issue, but you will find lot of things. If you click into all runs under the sample, you will see that they have 4 data. They have a 2 control sample and they have 2 treated sample, and you can see the accession number starts from DRR. Again, until unless you are satisfied with the data, you can look and you can choose and you can mention all the accession numbers which are present under the study all runs. That is how we select the data. So you can see that this one is having 15 runs, means 15 accession number. And what it is all about, again it is about something, okay, I have not read about that experiment. If I read it, it provide me the information on that CDK4 inhibitor dsRNA sensor for anti-cancer effects through the ER stress and immune modulation of tumor cells. And if you over here you can see that they have provided a pretty vast abstract information. If they have provided any GeoID, then you can look for it. So this is a one way of getting it, one way of scratching it and getting the data and accession number. So this is very important. Or in case if you are interested in for particular cancer, like for an example you are interested into, for an example, lung cancer, or could be a breast cancer. So if you are interested into lung cancer, you can search specifically for a lung cancer and you will find the data which supports lung cancer. So you can see that this is the RNA Seq live cell 3D models with engineered genetic subtypes of lung cancer. In study, if you click all runs, you will find the data which is like this: the mutated one, the control one, the vector one, everything. Up to here clear? Yes, you can use it, but the filtration which I have told you, yeah, we can use it, no issue with different platform, not at all, until unless that data doesn't have much difference, it's fine. Only the difference comes that whenever we are using a different organism, so we just have to make sure that the reference genome should be there whenever we are doing the comparison. Because for an example, if you are using a mouse data but you are doing you are first combining with a human data, then obviously it's going to give us an error. If not error, because there might be some similarity between the human and mouse, so there are chances that there are certain genes which we get might hit the human target, but still it won't be good, because we really want to find the genes which are expressed in mouse, not in human. I have just mentioned only the very basic filtration in over here. So if you are interested in using different platform, you can go for it, until unless you have a very good understanding, because in Illumina platform also there are different varieties of machines sequencer are present. MySeq, Nova, or everything comes under the Illumina only. And you can see that most of the times, 90% of time, people are using Illumina. So that's why I'm not changing anything over here. If you are interested into some more strategy, but it's always a good thing that you just stick to something which is similar like that. So public, RNA, and parent. Okay. So clear with this part? Okay. So now what I want is that you know how to do it, like pretty much clear with it. You will go to NCBI SRA, search for a data, and then you will use the filter which I have told you. Okay. Once that filter is done, you need to select a data. So I have selected one data. I thought of that it will be a good thing, but in case, if you, because I really want you to learn this thing, how to search for a data, because you might be interested into cancers, you might be having a specific cancer type or something like that which you might be interested in, right? Just make sure that once you open any data from the result, under the study, under the study you will select all runs, and then you need to note down all the accession numbers. Clear up to here? Clear? No need to, in strategy, no need to, I think so right now strategy is not. Just make sure that you are interested into RNA. So now, and that just be pretty much aware of it, that when we are going for human data or a mouse data, and you are very much interested learning this technology using human data, so that the size of data will be pretty big in size, that you need to be aware of it. Okay. Clear? So I have selected a very simple accession ID. Let's see that which accession ID you are interested in. So I've just selected uh, so this is the one which I'm interested in. Just a case, I'll show you that what exactly this is. Let me copy the bioproject ID, go back to the NCBI SRA, and paste this one, and you will see that it has 5 entries, in which you can see that WGS, that is whole genome sequencing, this is a DNA data, and you can see RNA Seq, so that is an RNA data. So what basically they did was they have studied the whole genome sequencing and RNA sequencing of myeloid cell lines. Okay, and they have used 3 cell lines. If I click into the show abstract, so they have performed whole genome sequencing on 3 myeloid cell lines. These are the 3 cell lines which they have used it for whole genome sequencing. For RNA sequencing, they have performed for these 2 cell lines. Okay, so and again this data is parent and they have aligned with this genome, human genome, HG19. So over here, if you want, you can again align with the another reference. That is up to you. I'll teach you that how to do that. This is just a data where they have both the things. Now under the study, if you click into the all runs, you will see that they have 5 entries and they have RNA Seq and WGS. So we are only interested into RNA Seq, right? So in that case, only these 2 accession numbers belongs to the transcriptomic analysis. So when I have to start my analysis, I will download this 2 data. Data 1 belongs to the cell line number MUTZ3 and another one belongs to MOL M-1. Okay, up to here clear? No, we are not going for whole genome analysis. The person who has performed both the things, whole genome sequencing also, just to find the variants, and also transcriptomic analysis. You can see that in that paper they have used both the things. In our case, we are only interested in transcriptomic analysis. So that's why we will take only the sample which belongs to RNA, not belongs to the DNA. Pretty much clear with the idea that how to search a data. So now what I want is that you might be looking for some other data, right? You might be having a lot of different ideas. So I want you to search your own data. Can you do that? Yes. So you're clear with all the steps, how to do it? Any issue with the steps? Okay. So I will stop here only. I will not go with the tools download and everything because getting a data is very important. So now today or tomorrow and tomorrow you will search a data which interests you and you need to write down the accession number you are interested in. Okay, today and tomorrow your task is you need to search a data in which you want to perform the RNA analysis. You will write down the accession number. Today is Tuesday. Thursday I will conduct your session. You need to tell me the accession ID you are working with right now. Right now, why I'm not sharing the recording with you, because I want you that you retrieve all the information, because I'm not going too fast. I'm just covering bits and pieces so that you can retain the information. For a later on, I'm recording everything. Even I will record a new sessions and I will share with you. So